My name is Christopher Fassbender, and I'm advisor to PETA Science Consortium International. I'll be moderating today's webinar along with Michelle Embry from the Health and Environmental Sciences Institute, or HESI, a nonprofit institution. And Michelle was the speaker at the first webinar in this series on weight of evidence approaches for bioaccumulation on the 10th of September last year. This webinar series is co-organized by the European Medicines Agency, HESI, the Japanese National Institute for Environmental Studies, PETA Science Consortium International, the US Environmental Protection Agency, and the US Food and Drug Administration. Please note that we will record this event and that the recording will be posted online. Please visit our website on the screen for the recording and for more information on future economics webinars. Please also note that if you write in the chat, not your full name, but only your initials will be displayed. The first two webinars in our series covered bioaccumulation and the Gill saline assay for acute fish toxicity. The topic of today's webinar is the use of in silico approaches to replace acute fish toxicity, touching to some degree also on chronic toxicity. Although alternative bioassays and computational approaches are available, the acute fish toxicity test is still one of the most frequently conducted aquatic vertebrate assays for chemical safety assessments. There will be two presentations today, each lasting about 20 minutes, followed by up to 5 minutes for technical questions. And after the technical discussion following the second presentation, we will have a general discussion. Without further ado, let's welcome our first speaker. To do this, I hand over to my co-chair, Michelle.
Thank you so much, Chris. Anna, would you like to please share your first slide? And I'll, while you're pulling that up, I'm very happy to be co-hosting this webinar series with Chris, and would like to introduce our first speaker. Anna Lombardo is a researcher in the field of computational ecotoxicology at the Mario Negri Institute for Pharmacological Research. Her main activities include data curation and the development of predictive models for environmental and ecotoxicological properties. And she's going to be giving a presentation on the use of QSARs and read-across methods for assessing fish acute toxicity. As Chris said, there'll be an opportunity to briefly ask clarifying questions after her talk, and please enter those in the chat. So with that, I'll turn it over to you, Anna. Thank you. And Anna, we can see your slides.
You can't?
Yes, we can see your slides, and I just unmuted you, so we're all set.
Okay, can you hear me?
Yes.
Perfect. So again, I will introduce today some tools available at the VEGAB website to assess the fish acute toxicity. These tools are part of NIES. NIES are new approach methodologies that allows the estimation, the assessment of risk and hazardousness of chemicals, reducing the use of animal testing. There are several NIES methodologies, in vitro, in ex vivo, in chemical, and the ones we are talking about today, the in silico methods. In silico methods take advantage of the existing data to fill the data gaps, to predict the missing values. They are cheap, fast. In many cases, they are free of charge and allows the processing of thousands of chemicals simultaneously. They are environmental friendly because they do not need solvents, they do not produce waste, and they do not use animal laboratory animals, so they have no ethical issues. In addition, these methods do not require the synthesis of the chemicals, so they can be used at the very early stages of the development of a new chemical. As I told you, All the tools I'm going to show you today are freely available in the download section of the Vega Hub website. In the news section, you can find a couple of ebooks and some videos that explain these tools, and in particular, how to interpret the results of these tools. All of the tools I'm going to present today have some common features. First, they are user-friendly tools. They need in input only the chemical structure as SMILES. SMILES are alphanumeric strings that represent the molecule, atom, bonds, aromaticity, etc., and that can be easily found on several public online databases. These tools are transparent and supply several information in the output. There is a tabular output that is particularly useful in batch predictions, and a more detailed output in PDF or in HTML, depending on the tool. Anyway, all these tools are not meant to substitute the human expert, but only to assist him or her. The human experts have to evaluate the results, to interpret the results, and decide on their own if they are sufficiently reliable and adapt for their purpose. The first tool is for read-across, in particular for the analog approach. Read-across allows the estimation of a property or an activity, like the fish acute toxicity, for a target chemical starting from one or a few similar chemicals, also known as sources. So read-across is based on similarity, and here is where the problems arises, because there is no universally accepted definition of similarity. These two are similar. The right answer is it depends. It depends on what you consider. If they are both mammals, they are more or less of the same color, they are both domestic animals, and both cute and cuddly. But so they can be considered similar. But if you consider their diet or their lifestyle, they are no longer so similar. In addition, read-across is usually made by human experts manually. So different human experts may have different backgrounds. I don't know, a chemist and a biologist have different backgrounds, may have access to different information, different databases, may give different weights to the same parameters. So they can produce different assessment for the same chemical. To try to solve these problems, we developed VERA, Virtual Extensive Read-Across, which automatizes the read-across in a transparent and reproducible way, including several similarity approaches and a mathematical definition of reliability. For a target chemical like the one in the box, for instance, VERA proceeds in with two different approaches. An unsupervised approach, so that do not depend on the property, that starts from the analysis of 12 entities that are chemical, physicochemical, biotic, or abiotic properties for 10,000 chemicals. These chemicals are divided into 2 or more clusters for each entity, and VERA project the target chemical, represented by the black star, in these 12 so-called subspecies. Then, in this way, is created a pool of possible similar substances on which VERA started the supervised approach. That is divided into 3 parts. The first is called VERA entities. VERA entities, all the 12 entities are weighted depending on the property of interest. And the weight of the entities and the similarity of the chemical substances selected are combined to give a prediction. The second approach is Vera alerts. Vera alerts start from the same pool of similar chemicals, but analyze them also considering the structural alerts and the molecular groups. Structural alerts are fragments, parts of the molecule that are associated with a certain activity, in our case, fish acute toxicity. In the output, you can see the 6 most similar chemicals that share the same structural alert and the prevalence of the toxic chemicals with the same structural alert. And a similar approach is used also to analyze the molecular groups. Then Vera combines this information, analyzing the co-presence of structural alerts and molecular groups in the similar, possible similar, to select the best similar chemicals and produce a prediction. The last step is the combination, the consensus between Vera entities and Vera alerts, and the calculation of the reliability that are reported here in the summary of the HTML output. The second tool is for QSAR. QSAR are quantitative structure-activity relationship, so they are equations that estimates a property or an activity relating the chemical structures and the activity or the property of interest. They can be quantitative in the QSAR, so they can give a number, 12 milligram per liter, for instance, or they can be qualitative in the SAR. In this case, they can give as output toxic or not toxic. To develop a QSAR model, we need a list of chemicals with 2 pieces of information. First, the property of or the activity of interest, for instance, 96-hour LC50 values for fish, well-curated values. And the second pieces of information are the chemical structures that should be well-curated and unequivocal. Using part of this set of data, we develop the model, and we verify the performance, and we define the applicability domain, the AD. The applicability domain is the chemical and response space in which the model gives a response, an output of a certain reliability. On the remaining part of chemicals that are unknown to the model, we test the model and the applicability domain. The tool is known as VegaQSAR. VegaQSAR includes more than 130 models for several properties: toxicological, ecotoxicological, environmental, physicochemical, or toxicokinetic. In addition, there is a tool that analyzes if each prediction for each chemical, for each model, is within the applicability domain of the model. On the VegaHub webpage and at this link, there are the QMRF, the QSAR model reporting format, documents that describe the model and that are mandatory if you want to register these chemicals, for instance, under the REACH regulation. For fish acute toxicity, we have 5 general models for fish. One of them is in classification, the other quantitative, plus 3 quantitative species-specific models, 2 for fetamino and 1 for guppy. But to assess fish acute toxicity, you may be interested also in other parameters, in other information, like, for instance, the fish chronic toxicity, or the mode of action of the chemical, or some physicochemical properties like the low KOW or the water solubility. And you can predict all of them at the same time using Vega. This is the PDF output. In the first page, there is a summary of the available information, the target, the prediction with its reliability, the experimental value where available, and eventual remarks or other information. The output is in a milligram per liter and in the measure unit used by the model. In another page, there is the list of the 6 most similar chemicals. For each of them, there are some information. The most important ones are the Vega similarity, which is a structural similarity, and the experimental and predicted value. Again, they are in the measure unit used by the model. So pay attention because they are not in milligram per liter, and you cannot compare the values reported for between the different models. You need to convert them using the instructions reported in the QMRF, or you can copy the smiles and predict them as a new target. We are working to solve this problem. In another page, there is the applicability domain index. It's an index that is spread from 0, not reliable, to 1, maximum reliability. And this index is made up by several parameters. Some of them are related with similar chemicals, other to the descriptors, and other to the structures like fragments, rare or unknown to the model. The list of parameters depends on the model because different kind of models use different parameters to assess the applicability domain. The third tool is Janus, that is a prioritization tool based on the PBT, CMR, and endocrine disrupting activity. PBT means persistent, bioaccumulative, and toxic. CMR, carcinogenic, mutagenic, or reprotoxic. Janus runs simultaneously 48 QSCR models, also available in the in the Vega QSAR tool. So why to use Janus instead of Vega QSAR? First, the prioritization. Janus gives in output also the scores that allows you to sort the chemicals from the most to the least hazardous one. Then Janus combines the available models, the available values for the same endpoint. In our case, it combines the 6 quantitative models for fish acute toxicity into a single output, and assign a reliability that is also based on other parameters like the water solubility. Janus also allows to generate and assess the microbial metabolites according to the rules developed by the EAWAG. On the other hand, Vega QSAR supply a more detailed output. So if you need more details, you need Vega QSAR. This is the output of the HTML output of Janus. Here, usually you have the list of chemicals, in our case, only one, the summary of the results with the scores. And clicking on the magnifying lens, you can see the details of all the models run for this chemical. In particular, for our case, there are the fish acute toxicity with their value and reliability as in Vega, the water solubility, and the overall fish that is the consensus with the value and its reliability. So to conclude, there are several in silico methods useful to assess the fish acute toxicity. In this presentation, I introduce 3 tools. Vera, which is a tool to automatize the read across, consider multiple approaches for the similar selection, and that also calculate the reliability of the prediction. Vega, that is a platform with several QSCR models that includes also an applicability domain index and the list of the 6 most similar chemicals that can be used to analyze the results and for the reasoning. And finally, Janus, that is a tool for the prioritization. It includes the consensus of the Vega models used in Janus, and also the possibility to generate and process the microbial metabolites. In the next future, this summer, will be also available CESAPI, which is a new tool for the assessment of the PBT and PMT properties of pharmaceuticals. I concluded, and I thank you for your attention. If you have any question.
Thank you very much, Anna, for that great presentation. Since we haven't received any written questions yet, so what we would do now is we would have time to answer any clarifying questions. Okay, I think the question we have so far, it's not really a clarifying one. So yeah, let's answer that after both talks. Yeah, okay, so let's move on. Please continue submitting questions anytime, and ideally it would be great if you could also identify which speaker you are addressing with that question. And with that, I welcome our second speaker, Paul. So can you please start sharing your slides? Dr. Paul Thomas is a European registered toxicologist with a PhD in aquatic ecotoxicology, and he's the president of KREATiS, a company specializing in high accuracy QSARs and other in silico models. He has more than 25 years of experience with industrial chemicals, agrochemicals, and biocides. Okay, with that, Paul, the floor is yours.
Okay, thanks very much, Chris. I guess you can all see the slide.
Yes.
I'm going to start just with one very brief one for those people who don't know me and don't know KREATiS. So KREATiS is a pure in silico R&D company, which is based in the southeast of France, close to Lyon, between Lyon and Grenoble. And our aim is to try to understand mechanistic interactions between chemical substances and biological matrices, and then be able to use that understanding to be able to quantify the impact on both human health and the environment. And we pass through phys-chem in certain cases, or certain parts of phys-chem, in order to be able to provide descriptors that we can help use in the models. We do both state-of-the-science modeling, which also now includes artificial intelligence, although that's not a part of the QSARs that I'm going to talk about today. And we also do eco-design modeling to help R&D groups to be able to home in on least or more safe and sustainable substances. And we produce bespoke in silico solutions for industry and for regulatory purposes. So we're now in our 13th year of existence. We have 15 multidisciplinary members, of which 8 are PhDs. So we have ecotoxicologists, toxicologists, organic chemists, machine learning experts, biostatisticians, all of which help our developer to be able to create quality solutions for our customers. We have also more than 25 years of regulatory toxicology and toxicology experience, because we worked on REACH dossiers, and there was a lot of recognition that the validated studies are really important to be able to produce quality QSARs. So I just wanted to give one slide on how we see NAMs, our position on NAMs, because there's a lot of controversy, I think, about this. Some people call it new approach methodologies, in which case is the high LL bioaccumulation test really a new approach. If I actually did my PhD on bioaccumulation on Acella aquaticus back in 1991, so looking at body concentrations in invertebrates is not really a new approach. Is new alternative to vertebrates also is the FET test, the OECD 236. Is that really an alternative to vertebrates? One might question it. Is this accepted in Europe, but maybe not everywhere in the world? And What about saying non-animal methods? Well, invertebrates are animals too, and they have a right to not be tested. So our position on NAMs, our ideal position, is really 100% animal-free, and it can be in silico, in vitro, from cell lines or other methods, which directly replace or contribute to the replacement of in vivo studies. And that means not just non-vertebrate studies or a reduction of the number of animals which are used, but as much as possible replacement. Of course, we understand that there are cases where that's not actually possible. And there will probably be, certainly if we don't know the structure of a substance, then we can't do much with it in terms of determining its potential toxicity. So then there are a need for other kinds of NAMs. And if possible, then a NAM must significantly reduce animal use. Let's face it, S9, for example, still needs a fish liver. So every now and again, you still have to kill a fish to get the S9. And when it is essential to use living animals, we can perhaps try as much as possible to use invertebrates as a sentinel species. And maybe a mix of all these three methods would help us to be able to cover the vast majority of the chemical domain. And to cut to the chase now on our model, what is it? It's the iSaferat toolbox. We call it iSaferat actually stands for in silico algorithms for environmental risk and toxicity. And we provide a desktop version of this. And the idea starts on the at 9 o'clock on the chemical structure, which is a SMILES code, which is then transformed into phys-chem properties, so particularly low kW water solubility. Vapor pressure is also useful for ecotoxicologists, especially if they're going to downstream do any actual lab testing on basis of this, because it helps them make the correct test design. And we use the water solubility as our main descriptor later on, at least for the ecotoxicity quasars. The next step, which is really important, is the creation of a specific MECOA subclass related to that chemical. And I'll talk more about the MECOA scheme in a minute. Then from that MECOA and the descriptor, we determine aquatic toxicity. That can be for algae, Daphnia for fish. We also do for microorganisms. We also have a published paper on how to do this for mixture toxicity, and it's not just an additivity approach as is used, by example, by the GHS CLP. We also have a biodegradation study, which is included within the software now. And on the human side, we can also cover skin sensitization, skin irritation, and dermal penetration, shortly acute toxicity estimation, shortly also endocrine disruption. We will have an endocrine disruption quasar at the moment for thyroid, and we're expanding on that. So go back to MECOA. What is MECOA? Mechanisms of toxic action. It's really equivalent to a molecular initiating event. It is not only that. Narcotics, for example, are not really what we consider a molecular initiating event, as they're just membrane destabilizers, but we consider a mechanism of toxic action to also include narcotics. Whereas modes of action are slightly different. They may be anywhere along that AOP, going from molecular interactions all the way up to population responses. So it's not quite the same thing. And what we wanted to do when we created MECOA was to use this method to provide a molecular initiating event upstream and a quantitative outcome downstream, regardless of whether that was going to be useful for ecotoxicological or human health determinations downstream with different quasars. So we can use this method for both human health and also for environment. Initially, we started with a model called MECOA, and now we've expanded on that with a lot of work together with Liverpool John Moores University, sponsored by Unilever, and that has now incorporated a lot of their work within our original MECOA scheme. And together with our own supplementary work, we've now created what we call MECOA Plus. MECOA Plus has just been included in the OECD quasar toolbox. So it's an add-in, you can add it in as a patch. And there are some publications on this. So Bauer et al. 2017-18 with the original ones on MECOA. There was one from Thurman et al. 2025. And I just had the good news yesterday that the most recent one, Levitt et al., was accepted in environmental science and technology just yesterday. So the idea with the MECOA scheme is it's divided into 6 specific classes. Those classes are going from least insidious to most insidious mechanisms of action, and with the smallest one being, or the first one being membrane destabilizers, and gradually working around through enzymatic hydrolysis, spontaneous reactivity, proactivity, where it's not the parent, but it's the metabolite that poses a problem, direct biological systems disruption, and finally specific interactions. So with each one of these is then divided into subclasses, and the point about these subclasses is that they're based on the mechanism of action. So the example I'm going to use here is the MECOA 3.1, which is hard electrophile activity, and in that case, we're looking at different substances here. So they could be epoxides, aldehydes, benzaldehydes. They have very different structures, but they have exactly the same mechanism of toxic action. And therefore, our point was that same mechanism, same effect downstream. And we tried this out, including it in our QSARs. The conceptual model of our QSARs is just simple linear regression. In this case, we use a thermodynamic method called subcooled liquid water solubility, where we take solid substances, we take a thing called the fugacity ratio in order to transform them into the solubility that they would have if they were actually liquids. And we plot that against the toxicity, whatever that may be, in log moles per liter. It could be acute, chronic, it could be fish, it could be Daphnia. And on this graph, you'll note there's also a dotted line diagonally. That dotted line is the solubility of the individual substances. So, and the solid line is the regression. So when that regression cuts through the dotted line, then that's the solubility cutoff limit, which means that anything beyond that limit to the left upper side of the graph is not soluble at the limit, is not toxic at the limit of solubility. So as we have one regression per MECOA subclass, or we're trying to produce that for all the MECOA subclasses, but there are a lot of them. We are now calling them QMARs rather than QSARs as quantitative mechanistic activity relationships. The next important point about the creation of these QSARs is that we spend inordinate amounts of time validating the data. We don't consider that the data is good as is, or is included in the disseminated database from ECHA, for example, in robust summaries, where they're considered CLIMISH 1 or CLIMISH 2 is valid. We revalidate all that data internally, and we find that sadly about 50% of that data is not valid or fit for purpose at all, and we therefore do not use them in our model. We include them in a master database where we explain why we're not using them. And this is an example where we've taken, if you want, the fish one for MECOA 3.1, which we've just been talking about. There was initially vast numbers of acute fish studies which were located for these substances, and for fish chronic there were quite a few as well, but we finally whittled them down. And the specific consigns which are included below, species, time measured and effect, are just the basics of what we look at when we validate these studies. We go into much more detail than that. And what you can see is that actually there are very few of the studies finally came out as what we considered fit for purpose for inclusion in our QSARs. It took us a long time for each of these subclasses to try to do that. This is what happens when you plot these very different structures, but they're exactly the same mechanisms on the same graph. As you can see, you get a nice mix with a nice regression line doing as we would expect. The mechanism of action is what is actually explaining the toxicity, together with the solubility as a simple descriptor. So once we know the mechanism, we've got the descriptor, we can quantify very accurately the toxicity. Today, accurately, this was a poster that came out from ECHA just in the recent CTAC in Maastricht. So ECHA had this on the first day, Advancing Aquatic Hazard Assessment, Curated Fish Toxicity Data and Benchmarking, and ISAFRAT was used here. And as you can see, the results were extremely satisfactory for safe rat models, between 84 and 94% within the accepted level of log units as they considered, with quite a reasonable number of data which were included in this case for the acute toxicity study to fish. This we can also push it a bit further. This webinar is supposed to be specifically about acute, but I think it is important to mention that it can also be used for chronic in the same way. This is the MECOA 3.1 training set with 9 compounds in it, and there happen to be a very nice spread across the descriptor, so we can get quite a reasonable degree of different types of substances falling within 3.1. And we look at specific statistics, the R squared, which is useful but not the most important aspect. It's the RMSE, which is the most important statistic here. And the smaller the RMSE, the better. So 0.27 in these cases are very good RMSEs. So how many can we do this for? Well, I'd love to be able to say we could do it for all of those subclasses that I showed you, but unfortunately that's not the case. It takes a long time to be able to cover everything, but we do cover major subclasses. So within the major subclass, I think it was about 70% of requests that we get from clients we're able to cover just on the subclasses we have. So we have 8 different acute fish MECOA subclasses, and 4 at this stage chronic ones. We're working on 2 other chronic ones. There's a lack of data for chronic, so it's very complicated to be able to find the appropriate data to do this, at least the quality data. What we do cover is non-polar narcotics, which cover a vast number of different substances that we encounter, including terpenes, ketones, ethers, alcohols, etc. We also have a good QSARs for polar narcotics, which include alkyl phenols, sulfonates. Also in the enzymatic hydrolysis, we can cover 2.1 hydrolysis to non-polar narcotics, which is the most important part of this MECOA class. That includes esters and lactones, for example. The 3.1 covers aldehydes, epoxides, the substances that I showed you in the previous slides. We also have the soft electrophile reactivity, which is the 3.2 that covers alpha beta unsaturated aldehydes like cinnamic aldehyde, but it also covers things like acrylates. And that these are, again, by far the most common groups which we find within the spontaneous reactives. Within the proactives, there are these are substances that can be detoxified in the case of 4.1 to narcotics, and we're trying to find a link now to see if we can actually use this to be able to, when we find such compounds, to be able to identify that it's perhaps more likely a narcotic effect rather than another kind of effect. And the 4.3, 4.4, we have been able to make that link. Within 4.3, we have substances such as eugenol, for example, and there are also redox cyclers in 4.4, where we have also created a QSAR for those. In the 5.2, it's acidification or alkalization of cells, so that could be carboxylic acids, but we can also expect to find bases such as amines, for example. Many amines would fall within this group, either 5.2 or 1.2, or what we call 1.2, 5.2, a double subclass. And now what is more complicated are when we get to the specific interactions. So these are for things like biocides, pharmaceuticals, and plant protection products. So we have certain potentials for nerve signal transmission disruption and ion channel modulation, but these are far from complete. We're still working on these, and we need to be able to create QSARs for those in the long term. This is a screenshot of our model. So as I said, we start with the Smiles code, which is put in here. Again, this is an aldehyde. This is non-anal. It'll give you the 2D structure in real time as you're creating the Smiles code, so you can check on the quality of Smiles code that it's putting in the right thing. Then it gives you the MECOA output for this. Then it will give you the physicochemical values. And you have the possibility of putting in a user input if you're not happy with the values that we're coming out with here. So it gives us, we have 2 log KW values. It selects the best one, which it then transforms into water solubility. And then this is what is used for determining the toxicity. The toxicity on the ecotox page then is put down fish, Daphnia algae, with the acute on the top and the chronic at the bottom. QMRFs are available now on our website. If you want to pull them off there, you can see what we can do in terms of QMRFs. While our QPRFs are currently only produced by our team, where we're not automating that at this stage, because if they're used for regulatory submissions, we want to be absolutely sure that they're 100% fit for purpose. I get this question a lot: do the authorities accept our QSARs? It's very difficult to get proof of acceptance because typically they are tacitly accepted. But I have had some indication from personal communication that they have been accepted. Many of them have been accepted, but ECHA never communicates that studies of any kind are accepted, only that they are not accepted. We haven't had any rejections since 2015, so we are fairly confident that they are being accepted. We have one demonstration of acceptance, which we have managed to locate. A client sent us this information because they sent in comments on a draft decision for a REACH dossier, and it was specified that the QSAR was used. I look at the Euclid dossier. The Euclid dossier has been filled in, and that chronic fish data has been accepted. So if you want to check that out, you can find that easily on the internet. Whoops, last point. Conclusions now is therefore that mechanistically based in silico models can replace in vivo studies, and they can obtain regulatory acceptance. Further, they can mechanistically explain empirical results, so they have more use than just a single empirical result that you don't quite know how you got it. High accuracy quantitative predictions can be obtained just from a single descriptor, in this case water solubility, when it's used in combination with a mechanistic methodology. The same methodology functions for acute and chronic studies, also for algae, for Daphnia, and for fish, and is being increasingly recognized by regulators. So I thank you very much for listening, and if you have any questions, please let me know.
Thank you, Paul, for that informative presentation. We have received some questions. There are no clarifying questions, but only questions related to the scientific or technical content. So now let's move on with going through the questions we have received. First, those on Anna's talk.
Chris,
Let's start. Yes?
really quickly, I'm sorry, there was one question that just came in that's, I think, more of a clarifying one for Paul on what the difference is between MECO-A and MECO-A+.
Oh, yeah.
And then and then we can kind of go through, yeah.
Okay, so happy to explain. So MECO-A was the very first one we came out. That was the publications Bauer 2017, 2018, and it was initially included within OECD Toolbox, and it was there until very recently, but just at the beginning of this year, we replaced it, or actually, I'm not even sure it's been replaced. I think it's been added to with the MECO-A+. So the MECO-A+, the original MECO-A one contained 60 structural alerts, and the MECO-A+ contains over 160 structural alerts. They are both included as freeware and freely available. We're also creating right now an API so you can pick them up off our website. And we're also working very hard on updating a thing which we call MECO-apedia. I perhaps should have put that on the slide. So if you tap in MECO-apedia into the internet, you will pick up our current documentation which explains the mechanisms, but this is about to be updated again sometime in this summer. We are going to be putting a much more extensive explanation of MECO-A+ in MECO-apedia, so you can get all documentation, all the references are in there. The other thing is that within our own software, which is commercial software, we also have a thing now called MECO-A Premium. So that's like the level up beyond MECO-A+. At this stage, it's not vastly superior, but we will continue to work on MECO-A Premium daily. MECO-A+, I hope we will put another one out there one day, but that's not going to happen for the next couple of years.
Thanks for answering that question, Paul. Then let's move on to the first question to Anna. How is the structural similarity calculated in the Vega QSAR tool? You can use different combinations of structural representations, for example, fingerprints, substructure counts, and of distance, for example, Tanimoto or dice. Which combination is used in Vega?
Okay, thank you for this question. The Vega similarity was published years ago. I will put in the chat the link to the paper so you can read deeply. But I summarize here the basic idea behind. It is based on a combination of molecular descriptors, fingerprints, molecular groups. They are all combined and analyzed under several point of view. So we can recombine them using a specific formula. Now I can put in the chat the link. Okay. I don't know if I answered or you need more details.
Thanks, Anna. The person who asked the question, you can follow up in the chat if needed. Appreciate it. So we're going to kind of pivot back and forth to questions that came up under Paul and Anna's talk. So this one is for Paul. For the iSafe rat tool, what kind of endocrine disruption properties can be predicted in the future? And will they be developed for targeting ED potential in humans or the environment? If you could talk about that and which species.
Yeah, at this stage, it's just humans, I'm afraid. We are trying to work again together with Liverpool John Moore University on trying to extend to environment, but there's not so much stuff out there on environment. So I think much of it will be done by extrapolation in terms of genetic similarity. But at the moment, these are human. So we have a thyroid QSAR, which was a project which was proposed by the NC3Rs in the UK. And that is on thyroid receptor alpha and beta. We're extending now to estrogen, alpha and beta. So what we're working on, and we'll have that within a few months. And then after that, we'll do androgen, which I think will hopefully be by the end of this year. And then we're trying to do progesterone. So the critical point is that we're using, now we've adapted a functional methodology through this NC3Rs project. We're able now to extrapolate that much faster on picking up the data. Again, the problem with the data is that there are a lot of false positives out there, and that needs to be eradicated before you can produce a decent QSAR.
Great, thanks so much, Paul.
Okay, the next question relates to the reliability score, and I think that goes to Anna's, relates to Anna's presentation. So is there increasing confidence in the output as the value approaches 1? For example, would a value of 0.85 be considered more reliable than a value of 0.30? And how do you calculate the reliability score? On what basis? And what is your definition of the applicability domain? I think all those questions belong together.
Yeah, thank you. So I start from the last one. The applicability domain is the chemical and response space in which the model gives a certain output of a certain reliability. In our case, the applicability domain is calculated through this index, the applicability domain index, that is made by several parameters depending on the models. And these parameters are combined to obtain a score from 0 to 1. So 1 is the maximum reliability, usually is assigned to prediction in which we retrieve the experimental value in the data set of the model, or with very high reliable prediction. Yes, 0.85 is much better than 0.3 as applicability domain index. Usually, we consider good reliability above 0.85, 0.80, more borderline. 0.3 is considered not reliable. But we usually suggest the user not only to trust this number as it is, but to examine the available information. Maybe that there are no similar chemicals, so the values of the applicability domain drops. Maybe the prediction for the chemical, that chemical is not so bad, considering the other parameters. So you need to examine it's the work that the user have to do. It's not difficult to obtain a result, but the difficulty is to examine all the prediction. I think I answered all the questions.
Yeah, you did. Anna, thank you so much. So the next question is focused, came up during Paul's talk, but I think both of you could probably speak to this a bit too, but we'll start with Paul. What are your most common exclusion criteria for the tox data that you spoke about, Paul, for the QSAR development? Like if you could speak broadly to that.
Yeah, we can't take nominal values, for example, as just being accepted. So it's not an easy case. In fact, this is why we go into more detail in terms of not looking just at the study in question, but also looking at the surrounding studies, because in many cases, nominal studies, for example, are being used for studies, especially in acute, but it's even occasionally occurring in chronic. And not necessarily for good reason. So as this can affect a test design, they've got then no idea about whether the substance was actually stable or not. But that's not an automatic reason just to exclude the study. So if we find that we have a study which is only based on nominal values, we then tend to look at the surrounding studies. We look at the physicochemical information. So, for example, is the substance likely to be volatile and likely to disappear? Did they use then a closed system, or did they use what kind of testing regime did they use? Did they use semi-static? Did they use flow through? This kind of thing. But if we can get enough information that suggests that this use of this nominal value makes sense, then and it was scientifically justified, then we will use it. But this is quite unusual, I have to admit. And most cases where you use that, you get that kind of information, then we are obliged to throw the study out. So that's a typical one, but it's a lot of it is in terms of test design. It's also in terms of test substance preparation. Certain studies have been carried out by using micelles or emulsions without necessarily even recognizing that fact. In certain cases, they find effects, but those effects actually turn out to be higher than the solubility limit if you actually really look into details about the genuine solubility limit of the substance. So there are a whole bunch of different factors that we need to look at on top of just the standard guideline ones in terms of whether fish the typical right species and what size they were and how old they were and how much water they used, etc., etc. So and the physicochemical parameters which are measured in terms of oxygen, pH. pH, of course, is a big issue within ionizing substances. So there are cases where the studies are carried out without a pH change, and the pH actually changed throughout the study. There are many things that we need to look at. But what I want to be really clear that the reason it takes us so long is because we don't just look at that one study. We tend to often look at the studies which are around it.
Yep. Thanks, Paul. And I had a follow-up, and I can let Anna weigh in on that. I'm curious with the criteria. So, we've talked about pros and cons of Klimisch, right, as kind of the standard. But for the work that you're doing within KREATiS, or maybe Anna, is there any sort of like standard template or capturing of kind of either whether it's a quantitative scoring or some kind of qualitative evaluation of the studies that could then be used and leveraged? And I'm thinking of kind of the idea of going beyond Klimisch and sort of getting at the details of why particular studies were evaluated or sort of how they were evaluated, why they were either discarded or included, that the broader community could use as far as kind of almost like a template for evaluation that's maybe a bit more robust?
There is one that's come from Ecotox Centrum in Switzerland. I originally worked with a guy there called Case, I think, but they've now changed. There's someone else who's just recently published something on this, where we're looking at a much more in-depth methodology for evaluating studies. I have to admit that if you're going to use this every time, it is extremely time-consuming.
Yeah.
And sometimes you don't need to go through all those steps to be able to decide that the study isn't valid, you know. But it is, if you're looking for something better than Klimisch, which is a little bit ambiguous, and I think many studies which are borderline Klimisch 3, i.e. not valid, technically not valid, which are perhaps the worst cases, actually have been slipped into Klimisch 2, perhaps by mistake. But this is. I think that it's between the Klimisch 2 and the Klimisch 3s, but some people seem to consider that just because it's GLP that it's automatically a Klimisch 1, and this is absolutely not the case. GLP is not a gauge of science or quality in terms of the science. It is a gauge of traceability. And I worked in GLP labs for many years, and it's a great thing to have, but it's absolutely not a proof of a quality study in terms of the science.
Yeah. Anna, did you want to weigh in on that at all? Okay. Just wanted to double check.
No,
okay.
I agree with that, Paul.
Thank you both.
Thanks. Then let's move on with data curation, which is necessary to develop good quality models, but it leads to a decrease of the number of data that can be actually used for QSAR development. So how do you perform external validation? How can we justify the use of these models in regulatory setting, considering the importance of the external validation in the OECD principles and the new QSAR assessment framework?
Can I answer that one, Anna, first? Yeah, so it is indeed a real problem. And this is why you noticed in MECOA 5.2 and 3.2, we don't have fish chronic. We have managed to get enough data to be able to create the acute studies, but we don't have the fish chronic in those two because there's simply not enough quality studies. It has to be said that these are MECOAs which are extremely difficult to test in labs as well. But there was a webinar that came out from ECHA, which specified the minimum number of studies that you could use in order to create a QSAR, and that is actually 5 studies, according to ECHA. They consider that anything less than 5 studies is not an acceptable QSAR. So we've got QSARs which are, they're not that low, but they're close to that low. And but the good news that ECHA also considered is that when you're using mechanistic approaches, you don't need to have a validation set in the same way that you do with statistical approaches, because there is a mechanistic explanation to the regression, and it makes you don't actually need to go in and then test it with a validation set. If you're using a different methodology, a statistical based methodology, then in that case you do need enough studies to be able to do a validation set. And that often you do see, I've seen quite a lot of articles where it's clear that in the validation set, the results are not as good as they are in the training set in terms of the statistics. So there is perhaps some overfitting due to that. But that's partly because you're not quite sure why those substances are in that group. As Anna said earlier on, it's an expert decision to some extent to include substances as similar substances or not. The approach that we're using is this mechanistic approach, where we're saying, well, the same mechanism of action, you will get the same effect. We've demonstrated that effectively, and that helps to increase the number of actual data that we have. So in 3.1, if we were doing just epoxides, I don't think we'd have enough data to be able to produce a QSAR for that, certainly not for fish chronic. But by having this mix with the different substance structures, you are able to increase that data set a little bit more.
Thanks, Paul. Anna, did you want to jump in on that?
We usually use also some, our models are basically statistical models, the majority of them. So we try to use all the techniques that can help when we have only a few data, because this is unfortunately a common situation, in particular for ecotox endpoint. And there are several techniques to try to solve the problems, like the cross-validation. So we do not have to divide the data set, the starting data set, in two parts, but we use one or a few chemical as a test, repeating these as many times as needed to have any chemical in the test and in the training. It's a sort of internal validation. It's not always sufficient, but it is a way to face the lack of data.
Thank you so much, Anna.
Yeah.
So there's a question that was maybe more specifically initially asked related to MECOA, but I think it applies to both of you. And it gets at sort of how this methodology could be applied for, say, nano scale materials. We've seen this a lot with kind of other sort of, I don't want to call them novel because they're not necessarily novel anymore, but different types of chemistries where our traditional, in air quotes, traditional QSARs may not be applicable. So I don't know if you guys could speak to that of, say, nano, or we might even have, you know, things like PFAS or other types of chemistries where some of these may not be as applicable. How would you approach that?
Okay, I'm thinking, sorry. We use sorry, can you repeat the beginning?
So it was really more of a question of sort of for these QSARs, where you might have compounds like nanoscale materials where solubility and partitioning properties and sort of our traditional ways of building these are not going to be applicable. How would you see the ability for prediction of these things and building of QSARs for those types of different types of chemistries? That was the question. And I was thinking you could speak specifically in the nano space or more generally to kind of the kind of chemicals traditionally out of domain.
Yes, for chemicals that are out of domain, you can try to build specific models, local models, with a few data usually that are available. So a module that focuses on that kind of chemicals. We face this kind of problems recently for pharmaceuticals, for instance. They are out of the domain of our usual model that are built on industrial chemicals because they are completely different as chemicals. So we have to work on them to build specific models, using specific approach for pharmaceutical, and the same can be done with the other group of chemicals.
Thanks, Anna. Paul?
From our perspective, right? Okay, so I have a position on nano. I really, at this stage, I'm not sure that even the experimental technology is there. I obviously there are cases where that it could be calculated, but typically it would be something like a silver nanoparticle or something, but it's basically silver, dissolved silver toxicity, which is causing the problem. So it's not really a nano issue in that case. And I'm not sure for organic nano what the issue would really be, but I don't feel confident at this stage that QSARs would have much meaning in that space. So, but on the other hand, there are many, and there are other areas where we know we're going to be constantly weak in the in silico space, and that's going to be UVCBs when we don't know a lot of the stuff that's in there. We have actually worked on a case with the UVCB where we don't know half the stuff that's in there, but as the other half that we didn't know were basically very, very high long carbon chain resins. We actually extracted them and considered them to be inert. We tested them at that inert fraction in a Daphnia magnia chronic study and found no toxicity. But we were able then in that case to be able to remove that from our in silico determinations, and we were actually able to get very good predictions compared to the laboratory results. So it's not all UVCBs, but if you've got a bunch of chemical structures which are actually potentially bioavailable, toxic in UVCBs and you've got no idea what's in there, I don't think QSARs are going to be that much help. So we're going to need other methods to be able to help you that. You mentioned PFAS, and I would also add organometals. PFAS, we've done some work on it, and actually it seems from our current work that most of the PFAS issues are related to the moieties of the PFAS, and they're not changing the mechanism of toxic action. So it could be that this method that we're using now works quite well for PFAS. Certainly that's the indications that we have. We're limited in the data, but it seems to be coming out that way. I would also like to see us moving to organometals, because I think this is an area where we could also be able to achieve something with this macro type approach. But there's not much data out there on that either. So if anyone has any data, we'd be happy to.
Yeah. Thanks, Paul. And thank you for, you kind of jumped in. There was a question on UVCB, so appreciate you touching on that.
Yes, I think we've covered already some questions that related to the applicability domains of the QSARs, including ISAF-RAD. So people asked, for example, about pharmaceutical application for pharmaceutical industry. I don't think we had that yet. So can you speak on that? Perhaps.
Anna, first.
Okay, yes. Not yet. We work on them in the last months, and we produce a new tool in which pharmaceuticals are assessed for ecotox part. But we also worked on not only on QSAR, that as Paul said, we have few chemicals, so they can be difficult to predict correctly. But we also work on read across, which starting from the similar, if you have similar chemicals, you can reason on that and produce something better than a model that is based on too few chemicals to be really predictive for the chemical. But I want to underline that in many cases, a model can be not good in general for a class of chemical like pharmaceuticals, but you have to analyze the prediction for your chemical, because maybe that for your chemical, that model gives a reliable prediction. So you have to analyze each prediction and combine multiple models, multiple approaches to obtain a better analysis of the possible toxicity towards fish in our case. So use several approaches and go deeper in the output of the tool.
Yeah, from our perspective, this is something that we're working on now. We actually have a PhD student who's working within a Horizon project called Pharma ERA, and she's doing ecotoxicity testing right now on a bunch of pharmaceutical substances. It's a huge task, of course, because there's a massive number of pharmaceutical substances out there, and she's just one PhD student, so it's really difficult to be able to, she's working like flat out. So this will gain some data for us and enable us to be able to produce certain QSARs for certain mechanisms of action on pharma. We also did a screen using the MECOA Plus approach to look at pharmaceuticals, and most of them came out with some kind of a MECOA answer. But our sense was that we're not certain of those results, because it may be many chemicals may have more than one mechanism of toxic action. It depends what's going to be in there. That everything will have something. It will at least be a narcotic. It could come out as a narcotic, but it's not actually a narcotic which is causing the problem, because it's a specific toxicity that we haven't been able to pick up yet. So as I said, this is an area we're now very interested in, and we're moving forward with this, but I think it's going to be a while before we will have real QSARs on pharmaceuticals that we can feel we can rely on and we would commercialize.
Thanks, Paul. Well, and I guess along those lines, there was a question that came in as far, and I'm curious about this, a question that came in on, have there been instances where QSARs have been accepted in the pharma space for ecotoxicity? And what you were just saying is that we need to collect more data and build those QSARs. So my assumption is the answer is no, but I didn't know if you had any perspective on that one.
My only perspective on that one, sorry, Anna, really jump in here really very quickly, is that I think this is a fantastic opportunity then for combining different methods, which may be in vitro as well as in silico, so that we get in many respects, the in vitro approaches can actually validate an in silico or invalidate it, depending on the case, an in silico model, whereas an in silico mechanistic model may empirically explain the results of an in vitro model. So these things would work hand in hand in this case. And I think that's probably the way we need to move in the future, not just reliant on any single one now.
Thanks, Paul. Anna, did you want to speak to that?
Yes, for pharmaceutical, okay, they are not accepted the prediction for QSAR, but we can use in silico methods to prioritize the chemical and identify the ones that are the most potentially hazardous one, and focus on that with the test in vitro or other kind of test that can be accepted. This is a first use of the in silico methods that can be already done. And we did it into a project named Premiere that focus on the ecotoxicology of pharmaceuticals. So we already work on this direction.
Thanks for that. Yeah, and along those lines, I know there's maybe just a couple more questions I see, Chris, but I had one, you know, in the realm of NAMs that we're talking about, and Paul, I think you did a nice job of teeing that up as well as you, Anna. And knowing that what we've heard is that for some types of chemistries, we don't have enough in vivo data to build QSARs, but in the future, when we don't do in vivo tests, how do you see kind of that either in vitro methods or other kinds of methods feeding into help with QSAR development for those cases where we really don't have that in vivo benchmark data? Do you have any perspectives? Yeah, go ahead.
Yeah. Already exists first attempt to model in vitro data. So we can start producing this prediction also for in vitro data and combine this information or try to work in the direction to complete the information. Not in vitro plus in silico combined together based on the few data available.
Yeah. Well, and I think the key is trying to kind of get the community involved in what types of data could be generated to help with that and bolster that. So did you want to speak to that, Paul?
Yeah, I think your key word is benchmark. But for me, it's really that. If there's some way that you can quantitatively benchmark in vitro outcomes, which would not be a direct replacement to a specific study, to in silico, and maybe it's a battery of studies, because if you look at studies such as the RTG, they don't cover, it's like everything, doesn't cover all applicability domains. So you have these different NAMs, which are some are good at some things and not so good at other things. And maybe we can create like the ultimate battery, and then we could bring that together with an in silico approach, which would actually be able to round up. I think from an ecotoxicity perspective, we're interested in being able to nonetheless quantify OECD 203, even though it's not environmentally relevant, it is still meaningful because we're talking about fish, or we're talking about Daphnia with the 202. So we're talking about organisms that actually live out there, maybe not in Europe, whatever, but they live in other countries, and they are therefore relevant. When we're looking at human health, I think it's more complex, because if you're using a rat to explain an impact on a human, then you're going from one species to another. But at least in the environment, we don't really have so much that issue. So yeah, I think there is a need to take into account that with the in vitro methods that we are going to be, which will be put in place, there's a whole battery of them, will perhaps be able to support in silico methods to provide an ecotoxicologically relevant outcome.
Yeah, no, I appreciate that. Thank you both. Just really quickly, because I know we've been talking around with both of this on kind of applicability domains, and I appreciate Marco asking the question again. Could you, Paul, speak a little bit more specifically on how the applicability domains are defined in iSafe-Rat?
Yeah, so in terms of the applicability domains, we follow all the ones that are required in the QAF. So there's a descriptor domain, a mechanistic domain, a structural domain, etc. All the individual domains which are required in the QAF are looked at. So we, within our model, will check off each one of those individual applicability or aspects of the applicability domain in order to come to a decision. And then, as I said, it's the team which actually comes to the final decision internally. So we will check using, first of all, that the relevant PhysChem data is being used, and that it may be that there are quality data out there which are actually experimental data rather than our predictions. So we would, in that case, we would rather use the experimental data. And then from that, we check that the substance falls within the descriptor domain, so which was in our case solubility, as I already mentioned. If we're outside of that space, then it's a kind of a judgment on how far we can go. If we're maybe half a unit beyond a long regression line, then we might say, well, okay, yeah, officially we're out of domain, but the reality is we're not expecting anything to happen on that regression, beyond that regression line, half a unit beyond where we already are. So we would consider that the result is nonetheless acceptable and will be fit for purpose. Even though we're not strictly within the applicability domain. So this is the kind of methodology that we will apply to all those different aspects within the applicability domain to come to a conclusion on whether we feel that a specific prediction is valid and fit for purpose for a regulatory submission or not. This is also, there's another point, it's important for us to be able to be in a position to judge that, and there are calls where we say, well, we don't think that this would get regulatory acceptance, and therefore we would propose a different method. So some of those methods may even be something like, well, we have significant evidence that the Daphnia are much more sensitive than the fish, for example. There are quite a lot of data that shows that, and therefore, even if we don't have enough data to have a fish model, we feel that if you can accept going with the Daphnia result, then that would be a conservative outcome, and that should be enough to be able to replace the fish study. So it's a little bit like some of the IATA approaches, which are being proposed in vivo, in fact.
Okay, thanks. I'll ask another question. How has the model performance in the QSARs that have been discussed been evaluated? So is it RMSE, R-square, or something else? I think that question can also go to both of you.
Yeah, it goes for both. I think, yeah. From our perspective, they're all important, but actually R-squared is not the ideal predictor of a regression. It's really a predictor of how well it is the toxicity is being related to the descriptor. So in fact, we have certain cases where actually the hydrophobicity, as that's really the surrogate for what we're using when we're using solubility, the hydrophobicity may not have so much impact on the toxicity. So it doesn't matter how hydrophobic your substance is, the toxicity is coming out very in a very similar level. So you'll get almost a flat line, a horizontal line. So there's no relationship between the descriptor and the toxicology or toxicity result, and therefore the R-squared is going to be abysmally low. A complete flat line would be an R-squared of 0. But so that's not necessarily, it doesn't mean that the toxicity result is not relevant or cannot be used. It can still be used, but in that case, the data that you need are the RMSE-based statistics and not the R-squared, because an RMSE is going to give you an indication of how close the data fit the line, and it's irrelevant of how well it fits the descriptor.
Okay, Anna, would you also like to speak on that?
I agree with Paul. He explained well. I can just add that these are statistics for quantitative models. If you have qualitative models, you have to use different statistics. like the adjusted accuracy or some other coefficient, the Matthews coefficient, or there are several statistics that can be applied in for qualitative models.
Yeah,
Thanks,
also within machine learning, there are different statistics again. So I was being very specific about simple linear regression here. I'd like to just point out. So it was not talking about other models where you may be looking at specificity, sensitivity, precision, etc., balanced accuracy as more useful indicators of statistics.
Thank you both. So there was one question that came in, which I think kind of gets to this sort of integration of different lines of evidence, not just relying on a single QSAR, about how would QSARs, or how might you address the issues that we are aware of related to species sensitivity, like the recent tire preservative and the different fish sensitivities to that? I don't know if either of you could speak to your thoughts on that.
Well, yeah,
both of you.
I'll start then. Yeah, that's a really this one is a very good example, and it appears to be salmonids which are particularly sensitive to this. But in fact, this was one of our early questions, especially in terms of fish species sensitivity. We initially looked at, our model contains multiple fish species, in fact, and we originally wanted to see whether we could find systematic differences. We were expecting, because everybody thinks that trout seems to be the most sensitive, of course, in the case of this PPDQ, it actually is, but it's not necessarily the case. And in fact, we could not find a lot of data out there which suggested there was any sensitivity difference between zebrafish, fathead minnow, carp, or trout, specifically in the regression models that we were using, and not even slightly so. Sometimes trout would be slightly more sensitive, slightly less sensitive. So there may be some really special cases like this one that you bring up, but I don't think it's systematic.
Thanks, Paul.
In our model, in our tool Vega, we have specific species-specific models, so one can also compare the results for different species and evaluate the different sensitivity. But in other new tools we are working on, we can also include the species in the input for the model, in the training set, I mean, and try to work on different species at the same time, but identifying the differences between the species. So this can be accounted. It's not yet in our tool, but in the next future.
Yeah, and I think to what you were both speaking to, I mean, the QSARs are one of the tools in the toolbox, and but they're not, the only tool. And I think the key is to kind of couple them with additional pieces of information. So that would address, hopefully, some of the species sensitivity. So I think we're at the end of kind of our list of questions. There was a request related to number one about some of the questions to Paul on the data curation, and if there's any kind of papers or resources that could be shared with this group, put in the chat, or shared with Chris and myself, and we could send around related to kind of those study evaluation template guides, things like that beyond Klimisch. That would be really, really helpful. There was also a question early on for both of you, if you have any resources related to kind of general training and education on QSARs as a resource for the community. I think some of us are probably, we know enough about QSARs to be dangerous. Others are just really kind of dipping our toe in. So if there are any from you both as experts in this space, general resources in the QSAR space that might be helpful, those would be great to share. And through the EcoNAMS webinar, we could probably make those available. Yes. So
may I say that we have a couple of ebooks that explain the very basic of QSAR available through the Vega website in the news section. So one can start to have an idea from these ebooks. And then, for instance, we organize courses. So
that would be great.
Yeah.
Yeah, and if you could send any resources, you know,
Yes,
links
I will,
that you have, you could either put them in the chat or send them to us. We can make them available. Sorry, go ahead, Paul.
So, yeah, we also provide training. If anyone's interested, you can go onto our website and you can see there's a page on training and what we do. And we've started now also by popular request to prepare a chemistry training, because like I'm a biologist and I kind of really wish I'd listened more at school, and I now to chemistry, because I really realize now just how critical it is in this area to really have a good understanding of chemistry. And many users are for like regulatory toxicologists or ecotoxicologists. They're like me, they don't necessarily have a very strong chemicals background. So we're now also producing a chemistry training specifically for QSAR use, because we by popular request.
That's great. Thanks, Paul. So over to you, Chris, to wrap us up.
Yes, thank you all very much for your input, many questions, and your participation. So we almost filled, I can say we filled the 1.5 hours very well, and we are grateful to Anna and Paul for their time and expertise. And so if you have any further questions, please get in touch with our speakers via email. Please keep an eye out for future webinars in this EcoNAM series. And after the webinar is closed, you will be redirected to our webinar series webpage. And there also will be a recording will be made available there. Okay, so with that, thank you all very much and enjoy the rest of your day. Goodbye.